It's really my great pleasure to share with this audience our latest work on studying the gene regulation in the 3D genome structures in the cancer genome. So I will start my talk today with this really oversimplified version of how the gene expression is controlled in the mammalian genome, right? So this green arrow can match to the direction of the gene transcription. So this is the transcription start site. So in the center of this picture, there's a promoter sequence whose role is to recruit RNA polymerase and initiate gene transcription. So far away from the gene promoter, there's usually an element called enhancer, whose main role is really to recruit tissue-specific or developmental stage-specific transcription factors. So enhancers are usually located far away from the gene they regulate, sometimes more than 1 million base pair away. But you can see from this cartoon, they can come over through pumpkin looping and further influence the gene transcription. Right? So how many enhancers are there in the genome? According to the latest research by the ENCODE Consortium, Nature 2020, by examining the ChIP-seq for different histone modifications and the open competence signals, we are able to identify close to 1 million such elements in the human genome. So this figure is from a couple of years back. So back in that paper, the authors have shown that back then there were more than 5,000 SNPs. They are related with different human disease or human traits, including cancer, cardiovascular disease. But one surprising finding back then is actually more than 93% of the non-coding SNPs that are related with human disease or traits, they are located in the non-coding part, and most likely in the enhancer or repressor regions, right? So a natural question for us to ask is how does this non-coding variance contribute to human disease? All right. So basically, the past decade or so, the major work has been really focused on the mutations or disruption of the enhancer sequence itself. I will use 2 case studies to demonstrate this point. So here's a Genome function. In this case, this paper was published in Science 2014 from the Rick Young and Thomas Luke group. So they sequenced like close to 20 acute lymphoblastic leukemia, pediatric leukemia samples. So they found this really surprising co-occurrent, this recurrent sequence here, in either insertion or mutation. They all form this consensus DNA motif that can be bound by this MIB protein. And they further show that once MIB bind to this newly formed DNA sequence, they can further upregulate the downstream TAL1 oncogene and further drive the leukemogenesis. So because this is induced by the mutational insertion, so we call it gain of function mutation. Similarly, there's also a loss function mutation. So this is actually one of the most famous enhancer example. This limb enhancer actually is located more than 1 million base pair away from its target gene, SSH gene, and actually is located inside the intron of another gene. But when the scientists delete this enhancer sequence, you can see there's severe phenotype. In this case, the mouse limb doesn't develop at all. So the mutations in the orthologous human enhancer sequence can usually result in the preaxial polydactyly disease, meaning you have a lot more fingers on your hand. Right? Again, the past research has been really focused on the mutation on the enhancer sequence itself. However, a few years back, this really groundbreaking paper published itself. Indeed, the concept is actually the enhancer hijacking can function as a new mechanism to drive cancer. So how does it work? So this is a normal chromosome 3, green bands gather 2, red bands is enhancer. They form chromosome looping events, and then they are far away from the oncogene, EBI1. However, in a big category for AML, leukemia patient, there's inversion going on. As a result, this enhancer is shuffled close to the oncogene nearby and then form chromosome looping and drive its gene expression. Again, the difference here is actually the enhancer is fine. There's no mutation for enhancer, but rather its location is changed and further contribute to cancer genesis, right? And then to study 3D genome structures of chromosome looping, people usually use HiC, right? Just to make sure we're on the same page, I'm going to go through the protocol for HiC. Of course, we all know the 2-meter long DNA need to be really properly folded inside this tiny nuclei, right? Usually in the scale of nanometers. You can imagine this is the chromosome looping events, 2 different strands of DNA tethered by this protein. And then in the HiC protocol, you first need to cut your DNA with 4-base cutter, and then you fill the end with this biotin marked DNA, induce ligations, and then in the last step, you need to share your DNA by sonication and to enrich your library with the biotin. right? So this is hopefully in your HiC library, you have millions of fragments like this. So one end coming from one location of the genome, and the other part coming from the other part of the genome. Right? So this is how when we process HiC data, usually we put them in the bins. Okay. Mindy, are we okay? Can you still hear me?
Yeah, now you're back. I was trying to type into you. Yeah.
Oh, okay. Yeah, because yeah, some strange thing pop up on my screen. Yeah. Anyway, so of course we can organize the HiC data in this data matrix. 10 here means you have 10 arrays from a HiC library. One end mapped to the 1K to 2KB region. So the other end mapped to the 6 to 6KB region, right? So of course, the bigger number you have, the more likely these two regions are located close to 3D space. And then of course, we can organize HiC, represent HiC as a heat map, right? And then the HiC map is actually symmetric. So usually that's why you see sometimes HiC can be represented as a square, sometimes as a triangle, right? So in the end, so this is the paper we published back in 2012 in Nature. So even if this is the first time we see a HiC map, you can appreciate actually form this kind of like triangle-like structures. So back then we called them topologically associated domains, essentially representing the hierarchical structures of DNA. And again, each dot indicating the prompt interactions between one region to the other region, right? So that's HiC back in Nature 2012. However, in 2018, by collaborating with my friends Jess Dixon and Job Dekker, so we showed actually HiC is really a powerful tool to detect structural variants, right? So how does it work? Again, this is a HiC map. Each dot usually indicates the interaction between one region to the other region, right? But suppose you have a translocation, right? In this case, you have, say, 10 KB fragments fall off the genome and then inserted to the other part. So of course, when this region is inserted, you can form local interactions, right? However, when you process your HiC data, you don't really have the cancer genome. You use a human reference genome, which is the wrong genome in this case. And then your HiC map will look like this. So it's this strange off-diagonal patterns. It's the base how we develop our algorithm to detect a full range of SVs, right? So here is a translocation. And in this case, of course, it's a deletion because you have this really wide space. And this strap is really between this region to this region, these crump interactions. And then when we can also see sometimes the butterfly kind of patterns in the HiC map. So this representing an inversion event as well. Right? Again, back then we showed HiC can be able to detect a full range of structural variants. The next question is, okay, now we have SVs in genome. How do we find hijacking cancers? So it turned out this question is pretty complicated. Just want to remind you that the cancer genome is really messed up. So in this case, you can see they have 4 copies of chromosome 7, right? You have really just messed up fusion events on chromosome 1. And then sometimes in the human genome, you can have this multiple band coming together, meaning the fusion coming from different parts of the genome. Right? So therefore, we developed this new loop finder. So essentially, it can really take care of all the abnormal fusion events in the cancer genome. The 3 main parts is using our software, we can really get accurate copy number variations in the cancer genome. And then according to different type and direction of structural variants, we can reconstruct the HiC map. And our software can also take care of the complex SVs. All right, so I probably will not go to the technical details, and you can find out in our paper. I will just demonstrate how it really works in the cancer gene misregulation. So this figure is generated using HiC in 4 bladder cancer patients. So they're all primary patient samples. You can, again, the y-axis, the copy number, 0 here is really 2 copy, and chromosome 1, 2, 3, 4, 5, right? So you can see genome-wide, seems like patient number 1 and patient number 3 looks okay. But when you look at the patient number 2, you can see actually this patient lose a big, the whole Q arm for the chromosome 5. Actually, loss of Q arm in bladder cancer has been reported back 30 years ago, one of the really prominent events. And when you look at the patient number 4, again, you can see a gain of copy of Q arm for chromosome 3 again, a big recurrent application in bladder cancer as well. And then we also use Hi-C to detect fusion events in pediatric brain tumor. In this case, again, this Hi-C map is between chromosome 12 to chromosome 8. Using our software, we're able to reconstruct the Hi-C map just for these parts. And then here's the fusion events. Then by collaborating with our pathology colleagues, we perform the FISH experiments. In this case, the green band marked the oncogene A2M, and the red marked the gene on this here. And then whenever you see this yellow spot, that means those are the fusion events that we can validate it by the FISH experiment. And then it really shows the high accuracy of SV detection by the Hi-C technique. Okay, so there are actually quite a few experts in this audience. For example, the organizer Kai Wang and Mindy, they're all experts on SVs. I hope I'm right in this case. When you usually have SV, right, you just have a list, breakpoints 1, breakpoint 2, breakpoint 3. But most of the time, you really don't know breakpoint 3 is connected with breakpoint 2. In other words, like you really use the breakpoints, you don't know the whole region, right? And this is really the key information for you to stitch multiple fragments together. So that's a part of an important module of our new loop finder, right? So essentially, in this case, we are just carve out the interchromosomal interactions. In this case, and more like a chromosome interaction for the cancer-specific alleles. And then we develop this principal component analysis based on the correlation by the rows or by the columns. And then by looking at the sudden change for the PC value, we can tell, okay, now we have reached the other point of the fragments and stitch things together, right? So next slide, I'm going to show you how it works. In this case, again, there's a fusion events between chromosome 9 to 22. Actually, thinking about it, this actually must be the Philadelphia chromosome in leukemia, which is actually the host for this conference, right? In this case, there are 3 fragments, and by using our software, you can really stitch out this evenly looking Hi-C map for 3 fragments and detect new tags or prompt looping events. Here's another example. Things are getting more complicated. There are many fragments in this case, A, B, C, and D as well. And then again, using our software, you can see by solely using Hi-C, nothing else, we are able really to get this nice looking Hi-C map and then predict the hijack enhancers, right? So we were able to apply our software in over 50 cancer cell line or primary patient. So that's probably a very, very comprehensive list about the Hi-C in cancer. Again, the X here, each data point is one cancer cell line or tumor, and the Y is number of new loops between enhancer or promoter. So you can see the top ranked the cancers, they're all breast cancers or prostate cancer. We agree with our known knowledge that they have more SVs. And the median new loops for the cancer genome is 37. So it's really, really a treasure for us for the explorer. Can you imagine how many of the key oncogenes or tumor suppressors are governed by these new loops, right? So in this case, I just gave you one case study. In this MYC, of course, we all know MYC is a notorious oncogene. In this case, this is a MYC is involved in enhancer hijacking in 3 different neuroblastoma cells. In this case, SKN-SH, SKN-AS, SKN-MC. You can notice that they're fused to different chromosomes, 8 to 7, 8 to 4, and 8 to 8. This is more like an intra-chromosomal translocation, right? In 3 different locations, MYC is involved with the enhancer hijacking, but the two different enhancer. So actually, in general, that's how we observe. We observe usually oncogenes are recurrently involved with the enhancer hijacking, but sometimes in 2 different enhancers. Most likely because in each patient or in each cell line, they have a different SV profiles. Right? Again, so this is a cluster analysis. Each column is a cell line. They're organized by their tumor type. The first 2 are prostate cancer, and then there's 4 breast cancer, and there's a big chunk of leukemia. Again, as we showed before, MYC has been really involved with enhancer hijacking in multiple cell lines. And in this case, for example, the prostate cancer, we see ETV1 and FOXY1 as well, right? Then how about their gene expression? In this case, we checked 3 cancer cell lines, leukemia, prostate cancer, and breast cancer. We compared the cell lines with the normal cell lines without SVs. So not surprising, actually, once the gene have the hijack enhancers, their expressions are higher than their expression in the normal genome without hijack enhancers. Right? Okay, so I think one of the surprising findings we have is actually by only looking at the genes involved in enhancer hijacking or new loops, we found a couple of genes that are sort of novel to the current literature, that their expression are related with the patient survival. So for example, in this case, in the acute lymphoblastic leukemia patients, we observed that UPK2 is really always involved with enhanced hijacking. And by checking the TCGA data, you can see the higher expression for UPK2 is associated with a poorer survival rate. And a similar case can be made for this COL828 gene in gastric cancer. Again, we just did a quick search, and we didn't see this thing known in the gastric cancer literature. So we have really a big supplementary tables. And if you're a cancer researcher, you're more than welcome to check the list. And hopefully there's something that can catch your eyes. Okay. All right, so once we make these predictions in these 50 cancer cell lines, 12 cancer tissues, we perform a series of functional validation. So in this case, again, this is a HiCMAP in HMAC, a normal breast epithelial cell lines. So this is TAT boundary. What we found is actually in T470, the breast cancer cell line, you can see there's a big deletion in this region, right? So this actually happens right next to the famous breast cancer gene ERBB4. So in the normal breast cell lines, this oncogene doesn't really talk to the downstream enhancers, probably because of TAT boundaries. But in the cancer cell lines, once the TAT boundary is deleted, and then my postdoc Jie performed a 4C experiment, and then it shows actually there's a prompt interactions between ERBB4 with the downstream clusters of H3K27 isolation enhancers, right? So that's just one evidence. We take this to the next step, because in the cancer genome, one copy has been deleted. So that's why we said, okay, let's target the same region. In this case, again, did the CRISPR deletion for the same TAT boundary, 88 base pairs, right? So once you delete the other allele, what will happen to our gene expression? Right, here's the results. By comparing the cancer with the normal genome, the ERB4 expression is increased from close to 0 to almost 5. But when we do the CRISPR deletion for the other copy, its expression double. So you can see like 0, 5 to 10, with one allele or both allele of the TAT boundary deletions that induce it to the enhancer hijacking. So in another case, this is actually a prostate cancer. Again, this is a fusion event between chromosome 7 to chromosome 14. In the normal genome, there's no chromosome actions because they have different chromosomal territories, right? But in the cancer genome, because these two parts are fused together, and then we predict that this oncogene is potentially driven or upregulated by the enhancer in the chromosome 14. And when we further zoom in, actually in this HiCBM, there are 3 enhancers. The second one actually overlaps with the exon, so we left it alone. So we perform CRISPR deletions for the first and the third deletion. Right? In this case, you can see here the results. Each bar is a one single cell clone, and then the Y is the relative expression. So the results is really, really exciting, right? By deleting a enhancer located on chromosome 14, we were able to reduce the expression for oncogene on chromosome 7, right? So without this software, it's really hard for you to imagine this could happen, right? So in our work, actually, we're able to identify hundreds of such genes and the genes and enhancer pairs. So if you are a cancer researcher, you're more than welcome to look at our list and really find whether your oncogene is potentially driven by the enhancer hijacking events. So with that, I can conclude our talk today. So first, hopefully I was able to convince you that HiC is really accurate in identifying CNVs and structural variants in primary cancer samples. And through pan-cancer analysis, we're able to identify enhancer hijacking events in 50 cancer cell types and 12 cancer types. And we discovered recurrent oncogenes. And we also showed that genes involved with the enhancer hijacking, they were generally overexpressed. And then we performed a series of CRISPR deletions. And we show that deletion of the TAT boundaries or directly deletion of the hijack enhancers can lead to the reduction of oncogene expression. So finally, this work was published, I think, last month or so in Nature Methods, and it was released our code, which is really freely available researchers, and you are more than welcome to talk with us if you have any questions. All right. So with that, I can acknowledge the people. So Xiaotao is really the person who designed the whole the algorithms and then follow through all the studies. And the other thing is Xiaotao will be on the market later this year. So if your department is recruiting, he will be a treasure to miss, right? And then GSU did the CRISPR deletion, and then Baozhen did the deletion for the hijacking enhancers. Jie did the deletion of the tag boundaries. And then some of my friends know that our lab recently moved to Northwestern. So this is Lake Chicago. This is the Lake Michigan. So this is the beautiful city of Chicago. There's still space on the boat. So if you're interested in joining my lab, let me know. There's still space for us to cruise. All right, with that, I can conclude my talk and answer some questions.